present. So fetal adaptations to adverse pregnancy is a topic I've been interested in for some time. Clinically, it's of interest because pregnancy problems are difficult to diagnose, and often the diagnosis is based on the adaptation that occurs in the fetus rather than a direct observation of the pathology that is affecting the fetus. Also, at SickKids and elsewhere, there's increasing numbers of interventions that are in various stages of experimental trials. And as you might imagine, there's a lot of anxiety about experimentation on pregnant women. And so that's one of the reasons that I've been interested in whether we can use model organisms and specifically mice to learn about the physiology. So the fetal circulation is very similar to the adult circulation, but it has some very obvious differences. The most important of which is that the fetus has a placenta, which is the source of oxygen and nutrients, and that connects to the circulation through the umbilical artery, which means that the umbilical vein enters into the fetal liver. The other sort of important differences are that the fetal circulation has a number of shunts. So the one is the ductus arteriosus that connects the main pulmonary artery to the descending aorta and essentially allows you to bypass the lung circulation. There's also a shunt called the ductus venosus in the fetal liver, which allows you to bypass the liver when needed. And then there's also this connection between the right atrium and the left atrium called the foramen ovale, which allows oxygenated blood coming from the placenta to stream across this connection and enter the left atrium into the left ventricle and go up to the brain. And so as I mentioned, the fetus will adapt under adverse conditions, and one of these adaptations, and one that is often used in the context of diagnosis, is so-called brain sparing, where the fetus preferentially routes blood to its brain and also its heart at the expense of other organs. And so we're interested in whether we could observe this physiology in mice and then start to probe it in the context of different adverse pregnancy conditions. And so clinically, you would observe brain-sparing physiology by Doppler ultrasound. And so as with humans, you can use higher frequencies in mice and do similar measurements. So here we're looking at the posterior cerebral artery. And then in this case, we've challenged the mouse by getting a pregnant dam to breathe different concentrations of oxygen. And you see changes in the blood velocity of the posterior cerebral artery in the fetal mice. And so this is, I guess, one of the adaptations that you would expect in a healthy fetus. And presenting that more quantitatively, on the left, we have a time course where we're looking at the mean velocity in that artery we were just looking at earlier as we cycle the gas challenge that the pregnant dam is breathing. And you'll see this sort of oscillating pattern as you move from 100% O2 to 8% O2, which would be very hypoxic conditions. And so you'll observe that the fetus initially compensates. So you'll see an increase in cerebral blood velocity, and then it decompensates. So you'll see a drop that kind of precedes the changing of the gas condition. And this corresponds to a drop in fetal heart rate. So you initially have a regime where it begins to compensate and then it can no longer compensate. And this appears to correspond to the saturation of the maternal blood. So as you put the pregnant dam under hypoxic conditions, the oxygen saturation drops, and then there's some threshold below which the amount of oxygen reaching the fetus is so low that it changes strategy and stops trying to compensate. We went on to look at this phenomena using MRI and specifically BOLD. So this is a T2-weighted MRI scan of a pregnant mouse. Unlike a human, there would be lots of fetuses in a mouse pregnancy. So here we're seeing one in a sagittal section where you can make out the spine, the placenta, the liver, brain. And so I guess one of the challenges of fetal MRI is also identifying all of these fetuses and appropriately setting up the scan geometry. So in this particular experiment, we did the same gas challenge that I described previously, where we cycled between 100% oxygen and then a very hypoxic condition, and looked at the bold signal. So this is T2 star weighted scans as an average in the brain and the liver. So this isn't functional MRI in the sense of doing a stimulus challenge and looking at specific brain regions. It's the overall oxygen saturation in the brain, in the organ that we're interested in. So you can see from this graph on the right that the fluctuations in bold signal associated with this challenge produce relatively modest fluctuations in the brain and very large pronounced fluctuations in the liver. And this was true across a series of animals that we examined. So I know that there are some noted experts on bold signal contrast in the audience, and they may be at this moment saying, well, wait a minute, it's not quite that simple. So of course, bold contrast is not a direct measure of oxygen saturation. So I tried to illustrate that with this cartoon, that if you have some gradient of saturated to desaturated blood, and the desaturated blood is more paramagnetic and thus produces a change in the susceptibility that perturbs the local magnetic field, you might get some inhomogeneous field pattern. Under hyperoxia condition, that gradient might be less, the resulting field pattern might be more uniform, and conversely, under hypoxic conditions, the variations would be more severe. And so the decay rate that you're seeing in T2 star weighted MRI is, of course, a function of that inhomogeneity of the field, and that translates into the decay constants that you're seeing. So it's not directly related to oxygen saturation, but closely linked to that. And so we, of course, the different organs have different vascular beds and different vascular densities. And so we went on to ask, could we model some of that in order to make our estimates more precise? So here's some simulation data. This was work done by Sharon Portnoy, where she generated distributions of random cylinders as a sort of surrogate for blood vessels with different densities, and then computed the resulting susceptibility maps and the associated magnetic field patterns, and then asked what the T2 star rates would be, and then worked that back into our bold calculation. So if you take that into account, the fact that the fetal liver is much more dense in terms of its number of blood vessels than the brain, then that disparity that we're seeing between the brain and the liver is less. But nonetheless, the data is consistent with this idea that there actually is brain sparing taking place in the fetus, where the oxygen levels in the brain are more preserved than those of the liver. So with this kind of observation in mind, we are encouraged to go on and ask about other types of things that we could model in mice. So the continuing with the idea that it would be interesting to look at hypoxia, we did an experiment where we raised pregnant mice in a hypoxic environment. So in this case, 11% oxygen. And we exposed the mice from 14.5 days of gestation to 17.5. So in this strain of mice, term would be 18.5. And 14.5 is the stage where the heart has its kind of normal allele conformation, aside from the presence of the foramen ovale, which I mentioned earlier. And so this would be simulating what you might expect if you had a placental defect that was depriving the fetus of oxygen in that kind of late growth stage of fetal development. So in this study, we looked at the mice with ultrasound and MRI. And then we also went on to look at them by micro-CT and by histology. So I'll start with some of the weight data. So The mice that were exposed to the hypoxia, the dams gained less weight than those that were in a normoxic environment. And this was associated with a small but statistically significant reduction in the weight of the fetuses. There was no decrease in the weight of the placenta. This is consistent with what we'd seen in a number of studies where often the placenta is normal in size despite effects that may occur on the fetus. But nonetheless, this was a model of growth restriction in late gestation. When we went to look at the placenta in these mice, we started by asking about the uteroplacental circulation. So this is the part of the placental circulation that is fed by maternal blood. So the maternal blood comes in through the uterine artery, which branches into these very long, spindly radial arteries, which join into these kind of oddly shaped spiral arteries, which then further merge into these canals before they enter the labyrinth, which is the kind of exchange region of the uteral circulation. And so using the micro CT data, which is of course three-dimensional, we could measure all of these arteries and then also start to ask what their like how well would blood flow through a circulation with that geometry. And so one of the things that we observed was that that there was statistically significant increases in the diameter of these radial arteries. And so these radial arteries are the primary source of resistance and also the pressure drop that's needed to ensure that the placenta operates at a low pressure. And so it's clear from this that the maternal circulation is adapting to increase flow through this exchange region in response to this hypoxic challenge. And so when we did computational flow calculations to come up with a total resistance for this bed, you can see that because the radial arteries account for such a large proportion of total resistance, that the overall resistance to the organ is greatly decreased in this exposure. From there, we went on to ask, well, what's happening on the other half of the placental circulation? So fetal blood and maternal blood don't mix, but they're brought into close proximity in this kind of labyrinth region, which is interdigitated by fetal capillaries. So again, we did X-ray micro CT to look at the 3D geometry of these placentas. And then from that, we used segmentation techniques that we developed in my lab to try and trace out all of the vessels, count all the vessel segments that make up those circulations. And so we observed that essentially the opposite happened in the fetus. So in the fetal circulation, you saw vascular volume was decreased on the fetal placental side. This was also reflected in a decreased number of segments. So on the maternal side, you have a circulation that's becoming more efficient, delivering more blood. On the fetal side, you have the opposite, placental circulation that's shrinking. And when you correlate those metrics such as the number of segments with the fetal weight, there's quite a strong relationship between the two. So in a sense, the placenta is on the fetal side is really only as large as it needs to be for the fetus that is being grown. So you could ask, well, which way does the causality run? Is it the fetus is building the placenta it needs or vice versa? But you can't easily answer from this data. When we look further at some of the hemodynamic measures, we saw things that we were familiar with from our clinical data. So when we looked at the velocity of blood in the ductus venosus, so this was one of those shunts that I mentioned earlier, that the bypasses the fetal liver. So you see a greatly increased velocity through the ductus venosus, suggesting that the liver is being deprived of oxygen and nutrients. Also, when we look at the umbilical artery, we're used to in healthy pregnancies to seeing a waveform that has a sort of pronounced systolic peak and then this non-zero diastolic flow, whereas that's largely absent in the hypoxia exposed mice, which is something that clinically would be associated with placental impairments or very unhealthy fetus. And so here's some of that data all consolidated in terms of the flows that we're seeing in different blood vessels. So we see an elevation of blood velocity in the middle cerebral artery, suggesting this kind of brain-sparing behavior that I described earlier. The ductus arteriosus is also elevated, suggesting that the fetal lung is being bypassed. As we saw just a moment ago, the ductus venosus is elevated, suggesting the fetal liver is being bypassed, and the amount of blood sent to the fetal lung is reduced. So essentially these fetuses, even though at this point the dam has been taken out of the hypoxic environment to the normoxic environment of the ultrasound suite, these fetuses essentially have a kind of baked in brain-sparing physiology. So just to remind you of this geometry, we're talking about more blood going through this bypass here at the ductus arteriosus, more blood going through this bypass here, the ductus venosus, and likely, although we didn't measure it, more blood going through the foramina valley and up towards the brain, where we see higher blood velocities. We went on to ask, well, so what are the implications of this for the fetus? And so using MRI, we measured brain volume, liver volume, lung volume. And as seen on this graph on the right, you have brain volume is largely preserved, liver volumes are reduced, and so are lung volumes. So this is essentially a model of asymmetric growth, as might be seen in, say, severe placental disease. We also went and did the challenge experiment. So here we have the mouse in their normoxic environment, and then we transiently expose it to lower oxygen and ask, well, what does the fetus do under those circumstances? So the ones that have been raised in this normoxic environment all along have the kind of healthy response of increasing their cerebral blood flow, increasing the ductus arteriosus, ductus venosus. So these two sort of brain-sparing bypass physiological responses are present. Whereas in the mice that are exposed to chronic hypoxia, the opposite happens. They don't sort of make what you would think would be the favorable response. They actually decrease their cerebral blood flow in response to a hypoxic challenge. So even though they have this kind of brain-sparing physiology baked in, they have no more reserve capacity to adapt. And so this might be one way that you can identify fetuses at risk, is that perhaps they've lost this adaptation mechanism. We also looked at postnatal development. So when we looked at body weights, they differed. Well, they differed by sex, as you might expect, but they also differed by whether they were exposed to chronic hypoxia. So mice that had been raised in this chronic hypoxia environment never achieved the sort of normal body weights. And then we also did MRI scanning, similar to what we've heard from the previous speakers. And so we looked at 2 time points, so P23, so 23 days old, which would be a sort of adolescent mouse, and then P59, which is an adult mouse. And so at P53, we could detect very subtle morphological differences, mostly in the posterior part of the brain and mostly in white matter structures. And at P59, we couldn't detect them at all. So it's clear that these adaptations that the fetus has work, in the sense that they do what they say and prevent large, widespread brain injuries. So I'm mindful of the time here. I want to contrast that with a different model that we've looked at, which is based on kind of late onset fetal distress. And so in this model, we create the fetal distress by prolonging pregnancy, and we do that by daily injections of progesterone. And so mice that are given this treatment don't go into labor, and so the gestation continues. And as you might imagine, that they keep getting bigger, and this becomes more a source of stress on them, and then at some point they're stillborn. And so you might think of this as a kind of late onset fetal distress. So when we look at birth weights in this population, you get, and we compared the fetal weights at different gestations, and then we compared the pups that are 1 day old to the fetuses that have had their pregnancy prolonged by 1 day. So they're the same age since conception, but one has been born and the other hasn't. And you can see that the ones that haven't been born yet are significantly growth restricted, in this case 14%. We also looked at the placenta in these placental weights in these mice. So the placental weight is roughly constant across late gestation, and then if anything, it decreases in that post-gestational or post-term period. And this is consistent with ideas in the literature that fetus, or so that the placenta has a kind of shelf life and that it becomes senescent. And certainly this was consistent with the data that we observed, and also some of the micro-CT results, which I won't show. So if you combine those two pieces of information together, the fact that you have a growing fetus and a placenta that is either kind of flat in its weight or dropping, and our placenta is our source of energy and our fetus is consuming energy, then the ratio of the two is kind of instructive in the sense that, you know, how much fetus is a given placenta supporting. And so as gestation progresses, that ratio rises and rises. And when you go post-term, it goes, you know, again quite a bit above the end of gestation value. So you can think, maybe perhaps the way to think of this model is that it's a fetus that outgrows its placenta, which it might be the case in late onset fetal growth restriction, which is a kind of acute condition where a fetus can go within a few days from being perfectly healthy to stillborn. So when we looked at these mice by histology, one of the questions we had was, is the brain and other organs hypoxic? And to do that, we used a technique called pimenidazole staining, where you eject the dam, and then based on the level of hypoxia at the time of when the mouse is sacrificed, you get a histological reading of which parts are below a certain partial pressure of oxygen. And so when we compared the percent area staining of a sort of normal gestational term mouse at 18.5 days to that 19.5 days, you see this dramatic increase in cerebral hypoxia. The effect is less pronounced in the fetal liver. So already at 18.5, there's some hypoxia staining, and that becomes more severe as you go 1 day post-term. So this is quite different than what we were seeing in the previous study, in that, you know, there the mice were brain sparing and largely succeeding at protecting their brains. In these mice, you have, you know, at least by this measure, evidence of severe hypoxia. So we've started to explore what are the morphological effects associated with that. So these are, I guess, two kind of contrasting models of how fetuses adapt under different conditions, and it seems to be very important whether, you know, the exposure is early or late in gestation. One of the theories of fetal growth is that a lot of the signals for how much the fetus should grow come from the liver, and so this brain-sparing phenomena of bypassing the liver also deprives the fetus of some of those growth signals, and maybe it grows less to compensate. Whereas in late gestation, you don't have the benefit of that opportunity to change course. So I'm just going to transition to, actually, I have one other slide on this topic. So, I mean, kind of projecting this to humans, so humans like mice have this exponential increase in their fetal weight as they progress through gestation. If you squint at this data, though, there is a bit of an inflection in the curve just at the end. And so it's been proposed that fetuses like our prolonged pregnancy mice eventually, or human fetuses eventually outgrow their placenta. And so that's one of the reasons that the growth velocity slows down, and perhaps as a signal as to when to be born. It's also become very much common practice to induce labor before fetuses reach 40 weeks, just to avoid the much increased risk of stillbirth in late gestation. Okay, so I just want to describe one other model that we've been looking at to try and explore this space, and that's a model of congenital heart disease. So arguably those others could be thought of as models of placental disease. Here we're trying to create a mouse model where the supply is normal, so the placenta is normal, but that the ability of the fetal heart to distribute that oxygenated blood to the right places is impaired. And so the specific type of congenital heart disease that we wanted to model is called hypoplastic left heart syndrome, where the left side of the heart doesn't grow as much, and so it very much has to rely on the right side of the heart in order to distribute the fetal blood. And so the way that we created this model is via an ultrasound-guided injection into the left atrium of the fetal heart at 14.5 days of gestation. So in this, here's a kind of schematic of the surgery on the left. So you have to exteriorize the fetus and then use the ultrasound to find the left atrium, which, as you might imagine, is extremely small at that age. Here's an ultrasound scan on the right where we're seeing the glass cannula and the fetal heart in a sort of 4-chamber view. If you put on your ultrasound glasses, you might be able to see the shear thinning biomaterial squirting out into the left atrium, but I will forgive you if you can't see that. So one of the kind of hallmarks of this disorder in humans is so-called retrograde flow. And so it's usually diagnosed on ultrasound in this so-called 3-vessel view, where you can see the ductus arteriosus or arterial duct, the aortic arch, and both in plane, and then the superior vena cava going through the plane. And so if you have a healthy animal, then the direction of flow through the ductus arteriosus and aortic arch should be in the same direction, i.e. towards the transducer in this view, which is why the color Doppler is red. And so referring back to our schematic earlier, this means that the blood is going from left to right in this view. So it goes up the aortic arch and then down the descending aorta, and also across the ductus arteriosus. So if you then go and block the left atrium, as illustrated by this X, then you no longer have blood going up the ascending aorta. In fact, you end up with blood going through the ductus arteriosus and then backwards through the aortic arch. And so that's shown up, or sort of illustrated by this red-blue contrast seen on ultrasound. And so you can imagine that this has an effect on how blood is supplied to the brain. Nonetheless, and it's actually very common in this population to see fetal brain injury or neonatal brain injury. Nonetheless, it's been very controversial as to why those kids have these injuries. And so, like I guess, prevalent assumption is that the genes that are important for heart development are also important for brain development. And so it stands to reason that any defect that might affect the heart might also affect the brain, and that would explain why you see this brain injury. And then, like, sort of competing hypothesis is that it's actually the supply issue that's the problem. That you have this abnormal blood flow, and perhaps that impairs the supply of blood, and that affects brain development. And so one of the reasons that we're interested in this mouse model is it kind of gets to the heart of that issue, because you have a model of hypoplastic left heart syndrome, which doesn't have this genetic defect. And so any effect you see on the brain should be a consequence of heart defect and not genetic effect. So we went on and looked at these animals. I'll just go through this briefly, but the hearts are very morphologically abnormal, as you might expect. So when we look at the dimensions of the ascending aorta, so these are MRI scans where we segmented the different vessels. You can see that the ascending aorta is much smaller when the mice are embolized. And conversely, the main pulmonary artery, which is the main vessel coming out of the right side of the heart, is much bigger. And so you have a mouse where you have a normal supply of blood, but it's being sent to the wrong places. And so we went on to ask, well, so what are the implications for the brains in these mice? And so we used the same deformation-based analysis that we saw in the earlier talks in connection with the cancer models, where we asked, what parts of the brain are either morphologically bigger or smaller as a consequence of this exposure. And we were quite interested to see that the parts of the main difference was a reduction in the volume of the part of the brain called the intermediate zone. So in the fetal development, the intermediate zone is the kind of future white matter of the brain, and is very often seen with lesions in newborns that have congenital heart disease and other pregnancy disorders. And so the fact that the mice would show this as well without having any corresponding genetic defect, we thought was quite interesting in terms of understanding the etiology of that condition. We also did the penminidazole staining that I described before, so there was some evidence of elevated hypoxia. We also did M.Eng staining, which is a measure of neuronal maturity, and so there's also some evidence that the neurons in these embolized mice were less mature than their sham-treated counterparts, perhaps suggesting developmental delay in this population. So there's a lot more work to do. It also, we think, is a great platform for exploring interventions that try and prevent these brain injuries. So with that, I'll conclude. Hopefully I've been able to persuade you that fetal mice can be used to model lots of interesting physiological adaptations that occur during pregnancy and which have some validity with respect to their human counterparts. As we saw, the prolonged hypoxia exposure produces some widespread changes, but the brain is relatively spared. This is contrasted with this kind of late onset oxygen insufficiency where the brain is not spared. And so with that, I'll thank many different people who contribute to this work. I'd particularly like to highlight the former research associate who's now a professor at Memorial University in Lansing-Cahill, and also the work of Anam Rahman, who led many of the studies that I just described, as well as our funding sources. So thank you.